In this video, I'm going to talk a bit about teeth and enamel in particular. So enamel is the crystalline part of the tooth, which is composed of hydroxyapatite, which has calcium phosphate and hydroxide in it. And I mean, this is kind of about digestion, so I guess to sort of get it out of the way, what is the point of chewing? Why do humans chew food? You could, a lot of people would probably think, well, it breaks it down and makes it easier to swallow, but we could just ask, why don't we just swallow things whole like a snake? And the reason for chewing, other than it does make it easier to swallow, look how snakes have to swallow things by unhinging their jaw and all that. But humans chew food. But one of the main reasons is that, say, we have some chunk of food here. When we chew it, it breaks into smaller pieces and increases the surface area of those pieces. And so digestive enzymes can actually get to it easier, and it makes it easier to digest things. So it increases the surface area. So if we look at teeth, we have the top part, which is the crown, and the bottom part, which is the root. The crown is made of the enamel, which, as I said, is that mineral, that calcium composed part of it, and that's the part that actually does the chewing. We have the dentin and the pulp here, and then of course the gums, which is what the teeth is sort of held in with. The bone, which could be the mandibular bone if it's the bottom jaw, or the maxillary bone if it's your upper teeth. We have the root canal, then of course the nerves and blood vessels, because teeth are living tissue. They do contain living cells. They're not like hair or something. And so let's talk a bit about these cells. So the cells that generate the enamel are called ameloblasts. And so the ameloblasts live in these stages, in these life stages. And so the first stage is the secretory stage, and it's secretory because they are secreting things. In particular, they're secreting these EMPs, which stands for enamel matrix proteins. So these are like a scaffold. So if you're making a building, you erect sort of a scaffold first, and then you can sort of build up the building inside the scaffold before then removing the scaffold. And that's kind of what these ameloblasts do when they're generating the enamel. And so I will sort of read through here and talk about it as we go. So the histological development of enamel crystals goes hand in hand with changes in ameloblast morphology. Undifferentiated epithelial cells receive signals to transform into secretory ameloblast cells of some 75 micrometers tall and 5 micrometers in diameter. And so that's these ones right here, which are these really sort of elongated, tall cells. And those are the ones that are secreting these EMPs here, these enamel matrix proteins, of which we will talk about here in a little bit. Amelogenin is one of the main ones. All right, so the 75 micrometer tall and 5 micrometer in diameter with specialized distal process called Tomes process. And that is this sort of picket fence looking thing here. So it's a process, not like, a way of doing something like, the process of evolution or something. It's a process as in something sticking off. And so those are called Tomes process, which plays an important role in matrix exocytosis. So exocytosis is the the process, I guess, using it in the other form of the word, whereby we have some vesicle coming down, and the vesicle comes and joins with the the cell membrane so that we end up with something that looks like this, and the contents can then come out of it. And so the contents of here would be that amelogenin that I was talking about. So those proteins, those enamel matrix proteins. These same cells will retransform into shorter cells, 35 micrometers tall. So that is these ones down here. So after these things shorten, after these ameloblasts shorten from the secretory stage, they enter the maturation stage. In maturation stage, ameloblasts undergo cyclical changes from a cell with a distal ruffled border, the ruffled ameloblast, or RA. So that's the RA up here. The ruffles they're talking about are these right here. To a cell with smooth distal border called the smooth ameloblast, or SA, which is this up here, where those ruffles smooth out. And then it talks about the tight junctions, so that's these purple things, and how those actually change position in these cells. And the organelle distribution differs. So we see that the organelles here are sort of distributed differently in the cell than they are in the maturation stage. And so what we see going on here, so we have these enamel matrix proteins coming out, and then the calcium phosphate, the apatite, the hydroxyapatite, which is represented by these hexagons here, will actually sort of start building inside these protein scaffolds until we start generating these enamel crystals, which make up the enamel of the tooth. And so we can look at this in a bit more detail. So this is the assembly of the amelogenin, which, as I said, is the main scaffold protein, or the main enamel matrix protein that we will be talking about. And so this is a single particle reconstruction of models. So the RM179 is the full protein, and that's what we have in A up here. Then the RM166 is truncated. That's what we have down here. And so the truncated version is missing the C-terminal domain. So these amelogenin proteins are comprised of three domains: an N-terminal tyrosine-rich amelogenin peptide, or TRAP, a charged C-terminal hydrophilic telopeptide, and so those two things are needed for forming these larger complexes. And then the central domain, which is the proline-rich region. So X and Y here can be sort of any amino acid, but there are these repeats of two amino acids that are proline and two amino acids that are proline, sort of over and over again. And so these are these proline-rich proteins that I talked about in the saliva video. And so what we see here, the truncation, in the full version, we have this right here, which is that C-terminal domain, which is missing in the truncated version. And what they show right here with these asterisks is these are the C-terminal domains, and so those have to come together in order to form this larger complex. So we see over here on top, we form these sort of these ordered structures here with the full amelogenin proteins, and then with the truncated version, it forms these sort of disordered, or non-orderly, I guess, sort of nanospheres. So these sort of aggregates of protein that are non-functional. And so we can look at the process by which this happens over here. In phase 1 here, we have the mixing of mineral ions, so calcium in green right here, and phosphate in red right here, and the full-length amelogenin monomers, which we have right here. So that's phase one. Then in phase two up here, at physiological pH values, the amelogenin oligomers in mineral pre-nucleation clusters are forming. So these are the mineral pre-nucleation clusters, and these are those oligomers. So these single monomers are coming together to form these shapes right here, and we can see that larger over here, and it has this sort of hollow part here. It's kind of donut shaped. And so in phase three, the amelogenin assemblies and pre-nucleation clusters then assemble into the proposed composite structures. So those the calcium and phosphate is sort of going inside those oligomers in phase three there. Then in phase four, protein mineral nanoparticles assemble into linear chains. So that's what's going on right here. This latter process will require some rearrangement of the amelogenin assemblies. Then in phase five, the linear protein mineral nanoparticle chains organize into parallel arrays. So that's what's going on here. Then in phase six, the pre-nucleation clusters fuse, leading to the formation of bundles of elongated crystallites of secretory enamel. So these are this is the enamel here, which has been secreted, and then the scaffold is removed, and we end up with just the hydroxyapatite there. And so that is the process of enamel formation. So now let's take a look down here at enamel degradation or demineralization. And so the demineralization process is this over here. So we have our hydroxyapatite right here, and we are in the presence of acid. So that is these hydrogen ions here, and it sort of breaks this single sort of molecular structure up here into three structures. So it's breaking it into three pieces. So we have calcium 3 and then phosphate 2, and we're losing a single calcium into solution plus water and some oxygen. Then we have our tricalcium phosphate here. We still have acid present. And so this actually breaks into two things. So these two single calcium phosphates here, and another calcium goes into solution. Then we can take 5 of those plus water and break that up into even smaller pieces and lose this phosphate here. And so the overall process is that the enamel is breaking down. And so what we can do, or what is done if we have like fluoridated toothpaste, or like here in the US we have fluoridated water, and you can argue about whether the government should be doing that or not, but the at least stated purpose of it is that the hydroxyapatite then turns into this fluoroapatite. So like this, so we have the hydroxyapatite here going to the fluoroapatite, and what we see is that the hydroxyl group here, or the 2 hydroxyl groups here, are being replaced with fluorine. So we see we have our hydroxyapatite here. We add this fluoride here, and then we end up with the fluoroapatite. And what's going on here, so when we have the hydroxyapatite, we will have those hydroxy groups, which are an OH like this. And in the presence of acid, we have these hydrogen ions. This oxygen will take that hydrogen ion, and we end up with something that looks like this. And this will be positively charged like that. And so then the electrons will move on to the oxygen there, and then we end up with our R positively charged and an H2O. And so what we do when we have the fluorine, and here I will do this one in green, is we have our R group here, which would be our sort of, calcium phosphate. So just kind of a generic R group. And then we have a fluorine. And even if we have acid present, this fluorine is not going to take on that hydrogen. So it does not become a leaving group. So not leaving a leaving group. And so that makes the enamel much more stable if we have that fluoride on there rather than the hydroxyl group. Because when that hydroxyl group cannot leave from here, then we don't get this breakdown that we see going on up here. And so it makes the enamel stronger and more resistant to acid. The acid, which comes from the bacteria that I talked about in the previous video, or acids that we get in our food, you know, for instance, if you drink soda or pop, as we call it here in Michigan, that is very acidic. Or if you have, coffee contains tannins, which are acidic, and a lot of our foods, are more acidic than they are alkaline, and so we can have a lot of acid coming in from our food consumption that can also break down the enamel. But anyway, that is you know, like I said, you can argue one way or the other whether the government should be putting fluoride in the water or whatever, but at least the stated purpose is that we have this going on, where we end up with the fluoroapatite like that. So this shows the hydroxyapatite kind of coming off here because of the acid, and then the fluoroapatite coming on here when the fluoride binds to it. And so, yeah, that is just a little bit about teeth. You could probably find, giant textbooks talking about teeth that would go into a lot more detail than I have. There are lots of papers that go into detail, of which I have quite a few linked to in the description down below if you are interested in learning more about this subject. But this is, kind of a broad overview of teeth and enamel in particular, which is affected by our diet. And that's kind of how I am justifying putting this into a nutrition and metabolism video series. But anyway, I hope you found this video helpful and interesting, and maybe it makes you appreciate your dental health a little bit better. But anyway, I don't want to keep blathering on, so I will see you in the next video.